Okay, so welcome everyone to the ARTnet update session of the 50th Annual Scientific Meeting of ANZSNM. I'm just going to flick to the QR code for people. If the QR code doesn't work, just enter in the 6-digit code which is on the screen for this session. I can see people are still joining the meeting, so I'll just leave this screen on just for another 30 seconds or so.
I can see Antoine online from Switzerland, so that's impressive.
Okay, so before we start the session itself, I just wanted to spend a few moments to reflect on the sad news that we received earlier this week on the passing of Sam Gambier. Many of us knew Sam either directly or by reputation, and Andrew Scott and Rod Hicks have kindly put together these words for us to reflect on. And the photo, which you can see here, is from when Sam visited us in Melbourne for the World Federation meeting just 2 years ago. And to me, this captures Sam perfectly as he was delivering us a very futuristic talk about the world of medicine. And we all offer our condolences to Sam's family and friends, are grateful to his enormous contribution to nuclear medicine. So again, I'm just going to leave this up just for a minute or so just to reflect on Sam's passing. Okay, so today's live session is on ARTnet, and ARTnet is certainly an undertaking that both ANZSNM and AANMS are very proud of how it's progressing. We do have a number of speakers today, and because of that we've actually pre-recorded the first part of the session. So I'm going to go straight into that pre-recording. At the end of the pre-recorded part of the session, we will actually be having an open discussion session, so I really hope everyone who's on the line stays for that, as all of the panellists are here to answer any questions that you have. So I'll be moving straight into the pre-recorded part of the session to hear about all the ARTnet activities and ARTnet trials. Hi, my name is Ros Francis and I'm the Scientific Chair of ARTnet. Welcome to the ARTnet session as part of the ANZSNM Annual Scientific Meeting 2020 virtual program. The outline of the session today will include myself giving an update on ARTnet scientific activities, followed by Michael Hofman and Louise Emmett providing an update on clinical trials in prostate cancer. Andrew Scott will then speak about the FET in glioma clinical trial shortly to be launched in Australia. Following this, Stuart Ramsay will talk to us about an Expect Quant Cardiac Amyloid project, which is part of a Siemens educational grant provided to ARTnet. Paul Thomas will then provide a brief update on behalf of the Executive Committee, and following this there will be time for an open discussion if anyone has any questions that they would like to pose. So ARTnet, what is ARTnet? ARTnet is a joint venture between ANZSNM and AANMS. It was launched in 2014 with a mission statement of aiming to promote and facilitate innovative collaborative clinical research utilising radiopharmaceuticals for imaging or therapy. And so the structure of ARTnet is that there is an executive committee which has representation from ANMS and ANZSNM and is responsible for strategic and financial decisions on behalf of ARTnet as a clinical trials group. Then there is the ARTnet Scientific Committee, which conducts the scientific activities of ARTnet. Olga Kovachev is our project officer. She's been the project officer for ARTnet since 2014, and Olga really provides wonderful service to us on behalf of ARTnet, and really we would not be able to conduct our activities without her assistance. So I'd like to pass on my gratitude to Olga for her help with ARTnet. The ARTnet Scientific Committee members are listed here on the screen, and there is representation including nuclear medicine specialists, physicists, radiopharmaceutical scientists, and nuclear medicine technology. And the ARTnet Scientific Committee is responsible for the scientific activities of ARTnet. And the committee meets quarterly and does a number of activities. This includes scientific review and endorsement of clinical trial protocols, advice and support for any ongoing clinical trials. And really the main focus of the scientific committee is to facilitate clinical trials, and also with a strong focus on quality. And this includes reviewing clinical trial protocols, which may be for proposed clinical trials or current clinical trials, or looking at various imaging within clinical trials. The scientific committee also has oversight of the camera validation program, radiopharmaceutical production and QC activities. And importantly, with the scientific committee, there is a strong focus in trying to link sites and really facilitate clinical trial activity by developing a network of research-enabled sites to really facilitate this activity throughout Australia. I just wanted to speak in a little bit more detail in some of the activities of ARTnet, in particular with some of the quality activities. So the camera validation program has now been running for a number of years now. So the program utilises an ARTnet Phantom to document camera performance for clinical trials. And the Phantom is sent out to sites, and this is coordinated by Olga and does require some degree of coordination. The Phantom is sent to site with a set of instructions. The Phantom is then acquired and the data uploaded to a core lab, which is outsourced by ARTnet, and perform the analysis. And then the analysis results are then passed back on to ARTnet, who then issue certification, providing the specifications have been met. And certification is isotope specific and for an individual camera, and it is valid for 2 years. Should be repeated sooner, obviously, if there's been a major software or hardware upgrade. Certification can be trial specific, so there may be certain requirements within a clinical trial that may require modification of the standard acquisition or processing, or otherwise there is a general validation which is to assess basic performance under standardised condition. Validations that have been performed over the last 12 months are of 12 different sites, and 16 cameras have been certified. So basically, Olga has coordinated sending out the Phantom. Most sites keep the Phantom for somewhere between 2 and 4 weeks, depending on how many cameras there are and what other things are going on at the particular site. We did just have one Phantom, but occasionally that does get damaged and is out of service, and so we've purchased a second Phantom to try and assist with getting a number of sites validated. There's been a strong focus on the upcoming clinical trials of ENZA-p, UpFrontPSMA and FIG, and we're trying to get those sites validated so that they're ready to commence those clinical trials. Just also I've put on this slide a reminder of the importance of this particular process within clinical trials, particularly clinical trials in a multi-centre environment. You need to ascertain that camera performance is within certain specifications, otherwise the data is just not comparable. And we really found this very much so when we commenced the ProPSMA study by assessing gallium-68 in a Phantom at the sites that were participating in the study. And as people will remember, we found a 15% SUV difference, which we just could not understand, and it appeared to be completely independent of individual camera manufacturers. And in the end, we found it was actually due to an error in a dose calibrator setting. But had we never performed this quality activity to ascertain performance, then we would never have discovered this error, and our SUV values would have been completely non-comparable. Internationally, there are other programs very similar to this, again, very important for clinical trial activity, the AANMS program and the SNMMI-CTN program, and we have discussions with both of these groups in the international area. I just wanted to touch on radiochemistry, and there has been quite a lot of focus within particularly the last few years, and more so in the last 6 months or so, with harmonisation of radiochemistry procedures and QC for clinical trials. This is particularly important when it's a novel radiopharmaceutical, and this is for either diagnostic or therapeutic radiopharmaceuticals. And harmonisation of the manufacturing and QC procedures within a clinical trial is actually essential in a multi-clinical trial setting, and ensures the quality and standardisation of the label and the final product for the clinical trial. So this facilitates the ability to run clinical trials where the product is manufactured on an individual site basis. And this is particularly important in Australia as we are very geographically separated. And that does create challenges, particularly with short half-life traces, which makes it very difficult to rely on a centralised radiopharmacy. So for us to be able to obtain enough patient numbers to compete internationally in these clinical trials, working together in a multi-centre environment adds to this. And as I say, being able to actually produce these novel radiopharmaceuticals on site does give us a lot more ability to be able to participate in these studies. Importantly, sites also gain experience in new radiopharmaceuticals using a standardised technique, and it is really very much contributing to the high-quality trials in Australia, promoting patient recruitment and leading to opportunities for clinical trial participation. In terms of the radiopharmaceutical looking at the radiopharmaceutical activities in clinical trials, ARTnet has participated in a number of levels. Within the scientific committee, we contribute to the development of nuclear medicine manuals, which outline the standardised procedure for the manufacture and QC, and this is developed in collaboration with trial investigators. Where there are relevant pharmacopoeia entries, these are used to guide the procedure manuals, and there is literature review and also wide consultation within our radiopharmaceutical scientists community within Australia who have been very supportive of this. ARTnet has also helped develop QC worksheets for validation runs and trial patient-specific worksheets. These have been employed within the ProPSMA and therapy trials and also will be used in the upcoming trials, which will be talked about, including ENZA-p, UpFrontPSMA and the FET and glioma trials. And these are very important for being able to ensure that sites are producing to the same quality standards across Australia. More recently, and really within the last 6 months or so, there has also been a focus on a new component to this, and this includes a site visit. And this is where sites are individually assessed to look at their radiopharmacy capacity, and I'll talk a little bit more about that. And then providing sites meet all of these criteria, then an ARTnet site certification is issued for a clinical trial with a particular radiopharmaceutical. So this is just an example of the typical validation run worksheet, which is required. This is the one that was used for the therapy study, and this is just showing the documentation for each particular run that is required, and then this is used just to validate that the criteria are being met for the production and quality standards of the product. So in the therapy trial, sites were required to perform 3 successful validation runs prior to certification, and there was a repeat validation required if there was any major change to synthesis methods. In addition to that, there was also a worksheet for every single patient dose in the study. For the upcoming trials, including ENZA-p and UpFrontPSMA, there is also going to be a site visit component required for certification prior to sites being open for the study. This includes an ARTnet-developed clinical trial radiopharmaceutical site accreditation checklist, and this is to ascertain the personnel staffing, facility equipment, process and quality, and clinical trial documentation within the radiopharmaceutical laboratory. So sites are sent this in advance. And then in addition to this, there is a site visit performed which was originally planned to be in person, but due to COVID requirements is currently being done by Zoom, where 2 people from ARTnet will visit a site and be taken around to view the equipment and the documentation which is available within the radiopharmaceutical lab. So this is just another level of quality initiative for multi-centre clinical trials. Following this, sites receive certification that this has actually been performed. And I'd very much like to acknowledge in particular Melissa Latter for her contribution to this. There's been a large amount of work to get this up and running, but I think it's incredibly valuable to ensure that the clinical trials being performed through ARTnet in Australia meet very high quality standards. We're very excited to see a number of publications now emerging in clinical trials for which ARTnet has been involved with. This is very pleasing to see, and it's really a testament to those investigators that have worked very hard to set up these multi-centre studies, and now these results are starting to come out. And importantly, these are high-quality publications. So Michael Hofman will talk more about the ProPSMA study, but we're obviously very pleased to see that that was accepted into Lancet earlier this year. And for those that are interested in citations, well, this publication's only been out a few months now, already has 15 citations, and we know it will be highly cited because the methodology paper published in 2018 has 50 citations already. And just to say that the management impact study, which was in fact one of the very first publications that came out for which ARTnet was associated with, really has been extremely highly cited, and was one of the top cited J&M papers in 2018, and in fact has already had 110 citations. So these are high impact publications because they are high quality research answering important clinical questions in nuclear medicine. This was just to show the trial that this is from the therapy trial, and thanks to Michael Hofman for this particular slide, but it shows that in terms of multi-centre trials within Australia, that there are a large number of sites that have been involved. This is covering a large proportion of Australia, and we're really pleased to see this because this was one of the real aims of ARTnet, was to facilitate linkages and to get these multi-centre trials up and running. We're very hopeful with future trials upcoming that will be additional sites that will be involved in those as well, and we do hope in the future that that will also include New Zealand to participate in some of these trials. That would be fantastic too. ARTnet is incredibly grateful to the support of other trials groups and organisations which have very much supported ARTnet and are working cooperatively and collaboratively in clinical trials in this particular space. Special mention to ANZSNM, who are really a fantastic clinical trials group for urological cancers, and we're working particularly closely with them with the prostate cancers, and TROG and COGNO working very closely with the FET and glioma study, and ALLG have been a great supporter of ARTnet as well. And so, really ARTnet is at its beginning, but is really showing great promise in the clinical trial space. Thank you very much. I'll now hand over to Michael Hofman and then Louise Emmett to talk about prostate clinical trials.
Hello, my name's Michael Hofman, and it's my pleasure to present to you updates on the ProPSMA therapy and UpFrontPSMA studies. I had the pleasure of recently presenting the results of the ProPSMA study at the EAU virtual meeting, the world's largest urology. Now, this is a randomised, multi-centre Australian study looking at PSMA PET-CT as an initial staging procedure prior to surgery or radiotherapy. This was really the very first ARTnet study. It was conceived by Ros Francis and I, together with nuclear medicine and then urology and radiation oncology colleagues from around Australia. And there was a grant call from the Prostate Cancer Foundation of Australia, thanks to an injection of funds from Movember. And we submitted this proposal thinking that, it wouldn't have much legs, and much to our surprise, it was funded. And this led to a large multi-centre trial, a really great collaboration of partners all around Australia: nuclear medicine, urology, radiation oncology, and importantly our biostatistics teams, trial coordinators, but also our radiopharmacy colleagues. And this was also done with the help of the ANZS Cancer Trials Group. ARTnet was pivotal in assuring high quality assurance both prior to the commencement of study and during the study. We devised a list of key specifications that must have been met by all study sites with regards to local production of gallium PSMA 11. And we also performed phantom images using gallium-68 filled phantoms, and we showed before study commencement that 10 of 14 PET systems actually underestimated the SUV by 15% on average, and eventually we identified this as an incorrect factory setting in dose calibrators. And this made me realise that testing the entire chain of measurements and calibration really should form part of a prerequisite for these multi-centre studies. The study really had amazing recruitment and finished almost 6 months ahead of schedule. You'll note in August 2018, we were randomising more than one patient every single day of the week, including weekends. And the results were pleasingly recently published in The Lancet. So if you have not read this manuscript, please follow the link and get up to speed. And also have a look at the online supplementary appendix, which is really has a rich data set with lots of interesting information. The primary outcome was that PSMA PET-CT was 27% more accurate than conventional imaging. That's an accuracy of 92% for PSMA PET-CT compared to 65% for the combined findings of CT and bone scintigraphy, and that included 2-bed-step SPECT-CT. We also reported the distribution of the PSMA PET findings and also key secondary endpoints, demonstrating change in management impact, lower equivocal studies, lower radiation dose, and, importantly, high reporter agreement. So in summary, the ProPSMA study demonstrates that PSMA PET-CT is a suitable replacement to current standard of care CT and bone scan. Pleasingly, this generated quite a bit of media attention when the Lancet publication came out a couple of months ago. So what's next for ProPSMA? We have a health economics analysis performed by our colleagues from CHIA group in Sydney. This is currently under peer review and will also be presented at the EANM meeting as an oral presentation and is a highly ranked submission. We also have ongoing follow-up of the men with PSMA negative PETs to define various time to failure parameters. We have tertiary analyses looking at PSMA PET compared to multiparametric MRI, or the value of PSMA PET findings within the prostate glands, and we're also looking at some artificial intelligence image analysis of these data sets. The experience with the ProPSMA study led us to believe that we could conduct these rather complex randomised trials in Australia with the assistance of ARTnet and clinical trials groups, and this led fairly directly to the evolution of the therapy trial. The results of the therapy trial were recently presented at the ASCO Clinical Oncology meeting, also a virtual meeting this year. And this is a randomised phase 2 study of lutetium PSMA 617 compared to cabazitaxel chemotherapy in men with castration-resistant metastatic prostate cancer. The aim of the study was to determine the activity and safety of lutetium PSMA compared to cabazitaxel, and we randomised 200 men around Australia to either lutetium or chemotherapy. And this was conducted at 11 sites around Australia. Importantly, we screened patients with PSMA and FDG PET, and only men with high PSMA activity at all sites of disease were eligible for treatment. The primary endpoint, defined by a PSA drop of 50% or more compared to baseline, was met with a 66% response rate with lutetium PSMA compared to 37% with cabazitaxel, a very large difference between the two arms with non-overlapping confidence intervals. We're also able to look at adverse events, and importantly, there were less severe grade 3/4 toxicities with lutetium PSMA despite better response rates compared to carboplatin chemotherapy. So the results of this trial concluded that in men with progressive disease following docetaxel, lutetium PSMA was more active than chemotherapy, with relatively fewer grade 3 to 4 adverse events and a PSA progression-free survival favouring lutetium PSMA. Again, lots of people to thank for this study, including funding from PCFA, Movember, It's a Bloke Thing, Can for Cancer, ANSTO that supplied lutetium-177, and Endocyte for supplying us with PSMA-617. Here's some nice footage of the therapy study team visiting ANSTO to learn a little bit more about how the no-carrier added lutetium is manufactured. Again, a big shout out to all the medical oncology, nuclear medicine and clinical trials teams around Australia that made this study possible. We recently held a webinar, a global knowledge exchange together with our partners at the Prostate Cancer Foundation. And remarkably, we had over 500 participants from 43 countries, all online, discussing the results of the therapy study. So what's next for the therapy study? Well, we await 170 progression events that will trigger the next planned analysis, and we anticipate another high-impact publication, possibly in the fourth quarter of this year. We've also got a rich image dataset, and we're interested in looking at the prognostic value of all these PSMA and FDG PET CTs. And we also took blood from all these men at multiple time points, which are currently frozen, and we will be performing a variety of translational endpoints looking at circulating tumour DNA and other blood biomarkers. Now, the third study we're going to talk about today has occurred as a result of another Movember-led initiative, which we were lucky enough to receive grant funding for. And our grant is called the UpFrontPSMA Alliance, using theranostics early to eradicate prostate cancer and develop new novel strategies. Study is the UpFrontPSMA study, a randomised phase 2 study of sequential lutetium PSMA and docetaxel versus docetaxel in men with metastatic hormone-naive prostate cancer. This is the team structure, another trial co-badged by ENZA-p together with ARTnet, led by myself and Arun Azad, one of our medical oncology colleagues, and a quick shout out to Knight, our nuclear medicine PhD candidate who's focusing on this project. So really, we've started with using lutetium PSMA as a last line of therapy after failing conventional treatments, and this study moves it back one big step to the hormone-sensitive group of patients and a group of patients with newly diagnosed prostate cancer. So this is the trial schema. We're taking men with newly diagnosed prostate cancer who have advanced metastatic disease on bone scan and CT and a PSA over 10. And then they undergo a PSMA and FDG PET. And if they have a high volume of disease on PSMA PET, they're eligible for randomisation to either the experimental arm, consisting of 2 cycles of lutetium, followed by standard of care docetaxel, or docetaxel alone. The primary endpoint of this study is undetectable PSA at 12 months and a number of secondary endpoints. This study is running at the therapy network of sites, and we recently randomised patient number 1 at Peter Mac in Melbourne, and we look forward to all the other sites opening in the not too distant future. ARTnet has been pivotal in this study, again, performing quality control and assurance activities before study commencement and during QC. And with Novartis taking over the intellectual property for PSMA-617, we have entered into an agreement with some slightly tighter quality control parameters than what we've done on the therapy study. All the PET images in our study undergo central review. We used the Widen system, a web-based system that anonymises the images and also checks the image quality before sending it out to one of 3 reviewers for central review. By way of summation, I'd like to highlight an editorial I recently wrote in the Journal of Nuclear Medicine after the publication of ProPSMA, a call out to the nuclear medicine community to change practices with prospective high quality data. And I wrote that establishing collaborative networks working together and upskilling the next generation of nuclear medicine specialists in clinical trial methodology are really key to achieving this goal. As new radiotracers emerge, there is a pressing need to develop and enrol patients in well-designed clinical trials. This is our best chance to properly evaluate the impact of our tests and enable widespread access by our patients. And I'd like to thank ARTnet and the leadership within the AANMS and the ANZSNM back in 2015 to establish this committee and the network. And a particular shout out to Ros Francis, the chair of the scientific committee, and Andrew Scott, who was pivotal early on in establishing this, but also our radiopharmacy, radiochemistry, nuclear medicine technology, our colleagues that make up the committee and enable it to function and succeed. Really a very big thank you to everyone involved. So none of this research would be possible without a large number of collaborators that really enable all of this to occur. So a shout out to everyone who's helped, both from nuclear medicine but also our uro-oncology, multidisciplinary collaborators, importantly our funding partners who really enable this work to succeed. Thank you. And lastly, I'll leave you with some footage from Peter Mac in Melbourne. Thank you.
It's my pleasure today to present to you the ENZA-p trial, which is a multi-site randomised trial funded by Movember and a collaboration with the federal government and run by both ANZSNM and the CTC's Clinical Trials Group with a lot of help from ARTnet. So the ENZA-p trial is really about precision medicine, targeting the right patient with the right treatment at the right time. The trial aims to use the PSMA paired theranostic agents, PSMA 11 and PSMA 617, to evaluate synergistic combination therapies in men at high risk of early treatment failure with single-agent enzalutamide, to develop and validate more accurate prognostic and predictive precision-making tools for metastatic castrate-resistant prostate cancer, and to use translational research to identify patterns of resistance with a view to developing more effective therapy combinations. There are a number of aims to the trial. The primary aim is the therapy trial, a trial of enzalutamide versus enzalutamide plus lutetium PSMA. And then aims B and C are translational and really look at developing predictive and prognostic biomarkers comparing PET and liquid biopsies. So what's the background to this trial? It's really started with these fantastic publications in mice and cell lines, and also early publications in humans looking at the upregulation of the PSMA receptor in response to androgen blockade. A small serial prospective study that we did at St Vincent's, looking at serial time point imaging, day 0, day 9, day 18 and day 28 following androgen blockade in men with both hormone sensitive and castrate resistant prostate cancer, showed something really interesting. The PSMA receptor intensity went down in response to hormones in men with hormone sensitive prostate cancer, but in metastatic castrate-resistant prostate cancer, there was this at times dramatic upregulation in the PSMA intensity in response to commencing either enzalutamide or abiraterone. This man in orange, you can see here, and there was a significant increase in both the number and intensity of lesions identified on PSMA PET between day 9 and day 0 on serial imaging. So what we interpret from that study was that PSMA response to androgen blockade is heterogeneous and that is dependent on disease stage and tumour genetics. PSMA upregulation is probably a marker of androgen resistance, and that combining enzalutamide with lutetium PSMA may be an effective synergistic combination in metastatic castrate-resistant prostate cancer. But most of all, that we need to know more. The PREVAIL trial was an excellent trial undertaken in enzalutamide in men with metastatic castrate-resistant prostate cancer and compared enzalutamide to best supportive care in 1,717 men. It showed a significant improvement both in radiographic progression-free survival and overall survival in these men. What's really interesting is when you look at the progression-free survival using PSA, overall, median PSA progression-free survival is 11.2 months. But when you look here, a significant proportion, about 40% of men, don't actually achieve that, and they fail enzalutamide very early. So what's the rationale for the ENZA-p trial? Men at high risk of early treatment failure with single-agent enzalutamide likely have multiple prostate cancer phenotypes, some of which are castrate-resistant. Adding enzalutamide and adding lutetium PSMA to enzalutamide may allow synergistic benefit, with the enzalutamide treating the androgen signaling inhibitor sensitive clones and the lutetium PSMA treating the castrate-resistant clones, and that combining treatments with synergistic value is potentially more effective than sequencing of these treatments. So the ENZA-p trial is an open-label, randomised, 2-arm, multi-centre, stratified phase 2 trial comparing activity and safety of enzalutamide versus enzalutamide plus lutetium PSMA in men at high risk of early treatment failure. So men must be early metastatic castrate-resistant with no prior chemotherapy in the metastatic castrate-resistant space and no prior enzalutamide. They need to have sufficient PSMA avidity on PSMA PET for enrolment, and they need to have at least 2 risk factors for early treatment failure on enzalutamide alone. There will be 160 men randomised in a 1-to-1 ratio to either enzalutamide or enzalutamide plus lutetium PSMA 7.5 gigabecquerels every 6 weeks up to a maximum of 4 cycles. So the primary endpoint is PSA progression-free survival, comparing enzalutamide to enzalutamide plus lutetium PSMA with a whole series of treatment response secondary endpoints. The imaging and biomarker study will involve FDG PET both at baseline and at PSA progression. We'll do serial PSMA PETs at baseline, day 14, day 96, and at PSA progression. There'll be matched pair circulating tumour cell analysis at baseline and in line with all the PSMA PETs. RECIST CT and bone scan will be undertaken 4 monthly, and there'll be 960 PET scans to analyse across 160 patients. Image quality and analysis is very important, and ARTnet's been very helpful with this. So there's phantom harmonisation of all the PET cameras and dose calibrators, accreditation and QC of all the radio tracer production. We'll be using MIM software quantitative whole body analysis with trial-specific harmonised workflows for both the PSMA and the FDG PET. There'll be centralised image analysis for screening, imaging, enrolment and serial time point PET analysis. And fortunately, there'll be automatic saving of all image quantitation up to a web-based server. This gives us wide scope for analysis of serial imaging using multiple methods. Looking at the translational study with imaging and biomarkers, so we're doing a matched pair investigation of serial PSMA PET, CTC enumeration, PSMA and CTC ARV7, looking at predictive biomarkers in a prospective well-defined cohort of men receiving enzalutamide. We're aiming to characterise biomarker findings that predict early treatment failure for both arms of the study. We'll correlate PSMA PET to other prognostic biomarkers in metastatic castrate-resistant prostate cancer, including FDG PET, CTC enumeration and CTC ARV7. And we'll look at PSMA intensity upregulation with a view to optimising combination treatments and guiding future trial design. So the impact of the trial will be to complete a randomised phase 2 study assessing the value of synergistic combination targeted treatments for men at high risk of early treatment failure, ready for phase 3 evaluation. We'll provide comprehensive comparison on the prognostic and predictive value and significance of both PSMA and FDG PET and CTC findings in metastatic castrate-resistant prostate cancer. The translation component of the study will provide insight into cellular mechanisms of resistance to both combination and single-agent therapy, a pipeline to increasingly effective combination treatments. And by evaluating these complementary synergistic combinations of treatments and targeting them appropriately to the right patient, we hope to make treatment more effective with minimal side effects so that these men can enjoy the highest quality of life for the longest possible time. Thank you.
I'll be presenting an update on the FET-PET in glioma or FIG study, which is a prospective multi-centre trial evaluating FET-PET in patients with glioblastoma. This is supported through grants from the Medical Research Future Fund, from the Australian Brain Cancer Mission through the Australian Government and Cancer Australia, and also the Cure Brain Cancer Foundation. It's being run through the Olivia Newton-John Cancer Research Institute, the Trans-Tasman Radiation Oncology Group, TROG, and also Cogno, and of course ARTnet is a pivotal partner in this study. So the FIG study is a prospective longitudinal multi-centre study. It's the first to pivotally examine the role of FET-PET in a multi-centre prospective way, looking at prognosis and also differentiation of pseudoprogression compared to recurrent disease in patients with glioblastoma after treatment. We're looking at a 210 patient cohort at 10 study sites across the country, distributed across most of the states of Australia, and also involving both major teaching hospitals as well as private practices, which reflects the radiation oncology treatment plans for patients with GBM. The trial sponsor is TROG, and the administering institution for all of the research funding is the Olivia Newton-John Cancer Research Institute. So just to remind you that the study objectives of the FIG study is firstly to determine the proportion of radiation treatment plans that are changed by incorporation of FET-PET imaging, and also after initial chemoradiation treatment to determine if FET-PET compared to MRI is able to more accurately differentiate tumour recurrence or progression from pseudoprogression. Secondary objectives are to look at whether FET-PET has prognostic capability performed immediately after initial surgery, and then to compare FET-PET parameters prior to chemoradiation therapy with other prognostic indicators which can be linked to progression-free survival or overall survival, to compare FET-PET with MRI, including perfusion-diffusion imaging, for sites of progressive disease after chemoradiation therapy, and importantly, so that we have information which can then be used for potential MSAC applications and MBS approval and health economic analysis as part of this study. There are exploratory objectives. Firstly, where FDG PET is performed in these patients as part of standard of care to compare FET-PET results to FDG PET, to correlate where the sites of relapse are compared to radiotherapy treatment planning volumes and how FET-PET may identify those areas, and also to correlate FET-PET with MRI and histopathology when subsequent surgery is performed after tumour recurrence. And there are a range of sub-analyses that we're intending to look at, which are quite exciting, looking at variability in treatment planning volumes as a consequence of using FET-PET, looking at the variability of treatment planning per se through part of the quality assurance and central reading process, looking at the spatial correlation between FET-PET parameters and also MRI and radiotherapy dose. And ultimately, we'll have a very rich data set where in 210 patients, we will have more than 400 FET-PET studies that we'll be able to analyse, and to be able to use various types of analysis approaches, including AI, to see if we can extract additional information that can be predictive and prognostic. The eligibility criteria are as listed. Patients must have good performance criteria, be more than 18 years of age or 18 years of age at study entry, histologically proven glioblastoma. MGMT status is not an inclusion criteria, and they must be suitable for post-operative chemoradiotherapy. They can have either standard radiotherapy, 60 gray over 30 fractions, or 40 gray 15 daily fraction course, and any type of temozolomide-based treatment can be implemented. It's taken us 12 months to develop the study schema and all of the parameters for credentialing of both the image analyses, the radiotherapy volume comparisons, and also the credentialing of sites. Patients will have FET-PET studies either initially after surgery. That will be a baseline study which can be used for prognostic analysis, or to see whether or not it would change the treatment volumes that are typically prescribed for patients undergoing treatment. 4 weeks after chemoradiation is completed, patients would have a further FET-PET study as a baseline. And then at the time of potential recurrence or pseudoprogression, a FET-PET study would also be performed. This is the only one which would be made available to the treating team. Management impact would be assessed before and after the FET-PET information is provided. The FET-PET itself will be through dynamic acquisition and creation of static images. There are credentialing requirements for all of the sites compared to a case study cohort of patients. NIM 7.0 will be used for analysis, and radiation oncologists will also, in a post hoc manner, assess the way that FET-PET will be incorporated into their treatment planning if it was available at the time that the original treatment plan was performed. And we will also be performing central and site FET-PET readings as well as for MRI. The FET-PET image acquisition will be through a list mode for 40 minutes with 2 statics being created. A benchmarking exercise, as mentioned, will be performed at all study sites before we commence recruitment at these sites. ARTnet is involved in credentialing of the PET cameras as well as establishing some criteria for sites that produce FET themselves for the study. Credentialing of the sites is underway, and we're very excited that the study will be commencing within the next 1 to 2 months. Thank you.
Hi everyone, I just want to give you an update on the ARTnet Quantitative SPECT-CT grant proposal establishing a multi-centre registry of individuals undertaking bone scans to assess ATTR cardiac amyloidosis. So those of you who attended this session last year will be aware that I presented then my proposal for quantitative bone scanning in cardiac ATTR, remembering that bone scans are accurate in diagnosing transthyretin cardiac amyloidosis. Effective treatment is now available for cardiac ATTR. The diagnosis using visual score is accurate but is not proven as useful in grading severity or monitoring disease progression, and that quantitation of myocardial uptake should help overcome these weaknesses. Single-site studies show quantitative SPECT produces meaningful SUVs using HTP and DPD in ATTR. So at that time, what was proposed that ARTnet was going to do in ATTR? Well, first of all, using a standard phantom with 3D printed inserts, we produced a phantom that mimics ATTR distribution in the heart. Potentially this can be transported around Australia to allow validation of XSPECT cameras for cardiac scanning. ARTnet was going to moderate a registry for XQuant SPECT of the heart for validated sites. Using standardised acquisition and analysis parameters, this would allow examination of diagnosis, prognosis and assessment of disease response. A call was made for EOIs for ATTR XQuant investigators. So what's happened since then? Well, we established an advisory team, myself, a nuclear medicine physician from Royal Brisbane, John Atherton, the Director of Cardiology and Associate Professor of Medicine at UQ, and Danny Liu, Co-Director of the Centre of Cardiovascular Research and Education at the School of Public Health and Preventative Medicine at Monash University. We thought about what database we wanted to use, so we need a secure, tried and tested database. We want to enter data at multiple sites and we want to keep the cost as low as reasonably achievable. So basically we decided that REDCap fitted the bill. A licence is held by Metro North. It's free to Royal Brisbane and Women's Hospital users, and we've obtained permission to use REDCap for this project. We then made a call via the ARTnet Scientific Committee for someone interested in assisting with REDCap database design, and luckily for us, Bridget Chappell has volunteered. We've also obtained permission to use the quality of life tool EQ5D5L for this project, and we've obtained REDCap subroutines from that organisation. We've written a protocol. That's pretty well finished now, and we've started ethics submission. So we've submitted the protocol to Royal Brisbane and Women's Hospital Ethics Committee. We think that approval at Royal Brisbane and Women's Hospital should make it much easier for other sites when they come on board. One problem we've found is that it's an ARTnet funded study, but ARTnet is not in a legal or financial position to act as a sponsor. So it's an investigator initiated project, multi-centre in nature, but with no external sponsor, Apparently, this is the first time such an application has been submitted to the Royal Brisbane and Women's Hospital Ethics Committee during the lifetime of the committee chair. So we're currently in negotiation regarding Royal Brisbane and Women's Hospital acting as the sponsor. And once we have ethics approval from RBWH, we will start contacting sites who expressed an interest. So what's going to happen in the next 12 months? Well, as we'd all be aware in the world at the moment, we're not really sure whether we're about to get extremely dark here in Australia or gradually it's going to get lighter and lighter. And I guess to some extent, whatever happens there is going to determine what happens with our project over the next 12 months. Thanks.
Hi, my name is Ros. Okay, so we're now back live, everyone. That was our pre-recorded part of the ARTnet session. I'm just, hopefully Paul Thomas is now unmuted because I'm inviting Paul Thomas to speak on ARTnet from the point of view of the Executive Committee of ARTnet and looking towards the future. So bring you on board, Paul, to speak to this slide.
Okay, thanks very much, Ros. And I guess it's my pleasure to talk about ARTnet, which has gone from nothing 5 years ago to pretty extraordinary now. And if you look at the number of participants that have logged on, there are 76, which I think is a reflection of the success of ARTnet, but also the interest of nuclear medicine research in Australia. And I think a lot of that has been, I guess, that incubation and that interest has been facilitated by ARTnet. Firstly, I would just like to thank everybody who is on the committees. They are, of course, all volunteers, and sometimes it is quite time-consuming. So thanks to everyone. Thanks to Olga, who's provides great support. I'd really like to thank Ros, who really has been the champion for ARTnet over the last 5 years and has taken us all along very enthusiastically for the ride. So thanks, Ros. Now, I guess the future of ARTnet is we hope to get much bigger, and one of the things we need to do, we've discovered, is that we need to look at the structure. And so it's been a little bit ad hoc, and now it's time to formalise some of those structures, particularly around the secretariat and the budget. At some stage, we will probably end up being a standalone incorporated entity. So that work is currently going on in the background, particularly the firming up of the budget is an important part at the moment. One of our ambitions is to align the scanner validation around the world, and we've had discussions with the AANM and the SNM, and they are also very interested in doing that. As you know, the EARL program has been around for a long time, and there are international trials that use the EARL program, and so it would be good if we didn't have to have different validations for different studies that we're doing in our department. So we are talking with the ANM about that, but things have slowed down with COVID. Ros talked about the collaborations we have with the other cooperative trials groups in Australia, and that's an area that we really want to push forward on. We'd really like ARTnet to be involved in all the cooperative trial groups that are using PET, and of course a lot more of them now are using PET in their clinical trials. So we'll be forging closer links with them. The EANM and SNM, it has been remarkable to witness over the last few years that suddenly Australia really is on the map for nuclear medicine research. And I think a lot of that's to do with ARTnet, also obviously the very high standard of trials that are coming out, but they're very, very jealous of what we're doing at ARTnet. And I think in the future, they'll be modelling some of their activities, and hopefully we can work much more collaboratively together. One of the things we're thinking about in the future is having a core imaging laboratory. So currently we have, I guess, different arrangements for each of the studies as to where the core imaging work is done. And there are some national initiatives around data storage and imaging laboratories that we will join up with. The other thing, and I think we've seen it already, is one of the briefs of ARTnet is to upskill the research and the activity of research in the nuclear medicine departments around Australia. So we certainly would like to encourage more departments to participate. And along with that, I think it also makes departments look at their standards, particularly around radiopharmacy. I think the radiopharmacy initiative over the last year or two has been really helpful. And it has enabled us to undertake some of these multi-centre trials that probably wouldn't have been able to be done otherwise. Having ARTnet validate radiopharmacies has meant that we haven't had to have GMP pharmacies manufacture some of these radio tracers, which would not have been possible. So I think ARTnet has made a huge impact already, and over the next 5 years, we're looking to, I guess, turbocharge ourselves. And, hopefully, Ros, you'll still be there at the front waving the flag for us.
Thanks, Paul.
Thanks, Ros.
Okay, so, well, thank everybody for their talks, and it was very good of every one of our panelists to pre-record them so we could just play through them. But this is designed to be an open session. I'm going to ask all of the panelists actually to see if they can put their videos on and unmute themselves. And please could any of the participants start either raising your hands to ask questions or enter questions into the Q&A box that we could actually answer. And I'll unshare my screen shortly so that we can actually see everybody's faces. I'll put my own video on as well, because as I say, this is in fact a live session, and we thought it's a great opportunity if anyone has any questions about any of the clinical trials, about the way ARTnet works, about any plans for the future, then we're all really keen to hear from everybody, and hopefully we can have a good discussion for the next phase of this. And I'm a bit of a newbie learning how to do all of the Zoom, and there are some questions that have been posted, so I might just start by stopping my sharing. And hopefully people can see all of us who are participating in the open panel. And as I say, if people have a particular question, raise your hand, and we should, as panelists, be able to join you on to ask some questions. So there was, in fact, a question from anonymous attendee. Does a patient on ADT for prostate cancer affect the image quality? So, Louise, you were the one speaking, I think, mostly about the effect of ADT on PSMA PET. Do you want to comment on that?
Yeah, so I think the answer is it definitely does affect not so much the image quality, but the result that you get from the scan. You do need to know whether a patient's on hormones or not at the time that they're imaged, and whether what state they're in, if they're in a hormone sensitive or a castrate resistant state. We've only done little studies to know what happens, and I think that both the UpFront study and ENZA-p will give us a lot more information on what it really means. And before that, I think there needs to be a lot more caution. UpFront's going to be quite interesting in the hormone sensitive space, because we'll get, Michael's already done a couple of patients, and both of those patients, I think, had a PSMA PET scan before they started on ADT and after they'd been on ADT for a few weeks. So we're going to get a lot of good information in prospective trials about what it really means. And in the meantime, I think we just need to be cautious in how we interpret.
Michael, did you want to add anything to that?
Yeah, no, I think Louise has summarised it pretty well. I mean, if you're on ADT and you repeat a PSMA PET, you're essentially doing a restaging scan. And you want to compare it to a baseline, ideally, before you're on ADT, so that you can see response assessment. But all our nuclear medicine tests are functional scans, so the appearance is altered by any active therapy. So you'll be able to tell to some extent whether your active therapy is effective or not. So it's very useful, but you need to interpret the results in the context of the current treatment and the clinical question being asked.
Okay, there's a couple more questions on the board, and I can see that some people have their hand up as well. So just from the Q&A, the next question is to Michael Hofman. How do PSMA 11 results translate to the often more or too sensitive PSMA 1007?
Yeah, this is a very commonly asked question, because there is now a little library of PSMA radioligands being used. It can be a bit confusing. There's the FDA-approved Axumin, which is fluciclovine, which is not a PSMA tracer, but sometimes confused as a PSMA tracer. That's an amino acid tracer. And then for PSMA, we have gallium PSMA 11. And then there are two main fluorinated tracers in use. One is F18 DCFPYL, and the other one is F18 PSMA 1007. There's a third one called PSMA RH PSMA, which is becoming available as well. And they're all pretty good. All these PSMA small molecules, whether they're labeled to gallium or fluorine, are extremely good. And but there are minor differences, and there's actually very poor head-to-head data comparing these traces because they've evolved in different jurisdictions. There's data that they're all extremely good, but not many people have performed a intra-patient study to look at differences directly between the two traces. So performing the same scan 1 week apart, this has rarely been done. We do know that the PSMA 1007 is a little bit less specific. So you see areas of benign bone uptake with a much greater frequency. There's published data on this. So it's a little bit less specific, but if you're aware of that, you can correct for that. So the German group that described this sort of false positive rate with 1007, cautioned it because, their expert reviewers reported them as benign. But if you get an inexperienced reviewer or reporter, then you may make mistakes. So you do need to know what tracer you're using when you report them. There's differences in uptake time between the tracer. There's differences in renal excretion. They're all pretty subtle. I think whichever one you can get your hands on will be excellent. There are some differences in intellectual property protection between the agents. Probably we're going to see there's an application for FDA approval for gallium PSMA 11 from UCLA UCSF, which is at a very advanced stage. And rumor is that will or may be FDA approved in September this year. Yeah. And that's an academic application. And if that happens, I think it'll result in gallium PSMA 11 being quite widely adopted in the US. At the same time, Progenics is putting together an FDA application for DCF-PYL, and I'm sure that will be FDA approved as well, probably sometime next year. And in Australia, there is an MSAC application that's just commenced for PSMA PET funding, and I hope that'll just be an agnostic application to let sites use whichever small molecule PSMA tracer they have available to them and encourage some competition within the market.
Paul, I was going to ask, I think you've done a bit of a head-on comparison, haven't you, with 1007 and PSMA 11. Do you want to make a comment on what you think about the bone lesions?
Yeah, so we've done a head-to-head comparison of 50, and it's certainly no doubt that there's more focal lesions that you see in the bones, very commonly in the ribs. And we've done all the SUV analyses and comparisons now, and it's clear that it needs to be fairly avid before you are going to call it a positive lesion, particularly in the ribs. And so it's going to come out to be an SUV something around 8. Wow. We hope to have all that data published soon. The other thing that you see, there is more inflammatory uptake. The ganglioneuronal uptake is much more prominent. I think part of it is related to the shorter positron range of the fluorine 18 compared to gallium. Some of those small things used to smear out a lot more, and now we're seeing them much more clearly. But I think once you get familiar and you're aware of the pitfalls, my feeling is that it's equally good. And the 1007 has the advantage of very little renal excretion. And so we've seen quite a few cases that we potentially would have called negative or have called negative on gallium dotatate, but they stand out very clearly on fluorinated PSMA 1007. So there are advantages for each of the traces. And as Michael says, whichever one you get is going to be much better than bone scanning and CT.
Or choline.
Or choline, yeah.
Okay, so we're just going to go back to the questions, and Kevin London asked a question about whether there are any paediatric trials on the horizon. And Kevin, I don't know if you're on the line that we can bring you on, or even if I'm putting Kevin on the line now. So if you want to join us, hopefully you possibly can unmute yourself. Look, this is a great question, Kevin, about paediatric trials. We don't have any currently through ARTnet, but we certainly would be interested in. all of the trials that we've run so far have had, ARTnet has facilitated them but has not individually run them. There's always been a PI, as you've seen and you've heard from some of the PIs of the trials. So we certainly would be very interested. We had some people interested actually with the upcoming FIG study. I have had some approaches from some paediatric units wondering about FET for paediatric brain tumours. But I guess we're interested in ideas. So do you have any ideas, Kevin?
Yeah, can you hear me, Roz?
Yes.
Yeah, I mean, really the thing that springs to mind is the therapy in neuroblastoma in the role of lutate versus I131 MIBG therapy, which, the standard old school I131 MIBG has had a bit of a rough trot with availability recently. And what my sense is, there's a bit of caution going to something newish like lutate for these sorts of cases, but there's certainly an opportunity there to explore it, and it would, if proven to be effective, it would really make a difference to availability of this sort of theranostic approach to neuroblastoma.
Yeah, I mean, we've got an interest in neuroblastoma at Peter Mac, and we certainly have treated a number of kids over the years with PRRT increasingly rather than MIBG, but I think there actually has been recently a drop-off in interest in the paediatric space because there was a phase 1/2 trial out of the UK of lutetium dotatate, which was read out last year as a negative trial, which has put us back a little bit. And I think our oncology colleagues are reading that paper and going, oh, these nuclear medicine people are just doing treatments, but when you study it properly, it doesn't work. I think it highlights the need that we do need to do these sorts of prospective trials, and I think increasingly our paediatric oncologists will not refer us patients just for compassionate access therapies. They'll want it to be done as part of a clinical trial.
I mean, that's so important, that's so important that all patients we see, should be on a trial, much like how paediatric oncology has sort of come forth in the last 20 years, where every patient has been on a trial. Pretty much, that's been the focus, and I think we need to do the same thing. And where there's such a glut in evidence, that one trial has really had an impact in sort of being the paper that people quote when they're thinking about it, and it makes a worry when there's only one paper. It has to be validated. And we need to sort of look for opportunities.
We need to be writing more grants. What we all need to be doing is finding funding and taking every grant opportunity and trying to put that into the nuclear medicine space. It's a painful thing to be doing. No one likes writing grants. No one likes writing grants that don't get funded, but it's the way forward.
And in the paediatric space, there's actually quite a lot of funding opportunities. So the percentage of applications that are actually successful is a lot higher than you might think.
So it sounds like
you just need to partner with a paediatric oncologist and apply for the next grant opportunity, and you might be surprised, but if you drove a project from an academic perspective, I think you'll find there will be sites all around Australia interested in following your protocol. And a group like ARTnet, probably together with perhaps a paediatric oncology trials group, can do this pretty successfully in Australia.
Yeah, I agree.
So I guess that's an encouragement to anybody out there who's got studies. I mean, Kevin, has raised the paediatric question, but there are a lot of really interesting questions out there. And I think, one thing about ARTnet was the whole aim was actually to be able to facilitate these kind of trials and make them a little bit easier to do. So, and also with the backing of a trials group, we've certainly found the chance of grant funding success does go up. And because you have a structure around you, which gives the grant funders a bit more confidence that the trial is actually going to happen. And so I think that is something which surprised all of us in the beginning. I think Michael commented in his talk when we put in our first grant, we were incredibly surprised to get shortlisted. But I think it was because having the trials group backing behind and having a system really does help. And these questions are important to answer in a clinical trial setting, and they can be done. So I think it's been great that so many people have put in for these major grants and have got them, but there are opportunities. So we encourage people to do it. Okay, there are still lots of questions, which is great. There is a question, multi-point actually, from Judy Diong, just asking about involvement in the clinical trials and how sites are selected, and whether sites can join trials that are open, and some questions about launching research trials through ARTnet, and also a question about ARTnet certification. So, I mean, I guess I could go to either Mike or Andrew even, well, to talk about how do sites get selected in the trials. Well, I can answer it from an ARTnet perspective. That's down to the principal investigators of the studies. ARTnet doesn't per se select the sites, but ARTnet is really happy to connect interested sites with the principal investigators of studies. And usually sites are selected based on the patient cohort and the expertise at the site. But I know Andrew has just gone through this for the FIG study. I don't know if you want to briefly touch on the process that you went through to select sites, and then I can ask some of the others to comment as well. But it's super great if this question is because sites are interested, then that's what we want to know. So we would say, please do connect with ARTnet and we'll try and connect you to the investigators. But I'll hand over to Andrew to talk about how that site selection process happens for multi-centre studies.
So it very much depends on the protocol. So if you've got a somewhat complicated study like, the FIG study or the UpFront or the ENZA-p, which is multidisciplinary, and where you've got multiple cooperative groups involved, you will need to go through a site selection process, which is fairly standard, where you actually send to key people at major institutions, usually through the cooperative groups, and ask for expressions of interest. And then you have a process to actually assess whether the sites are capable of actually doing the study. And you know how many sites you've got a budget for that's consistent with the number of patients that you've got to recruit. At a more general level, if you have, however, a study similar like the SPECT project, which was mentioned by Stuart, that's more of a investigator-initiated, more cooperative within a nuclear medicine community-like study, where it's much more likely you can have multiple groups involved. So it depends on the study, and it depends on the questions which are being asked and answered.
Yeah, it's also one comment about multi-site trials is you have to pay a per site payment from your grant. So if you have a large number of sites involved, it can become very, very expensive. So it's great to have lots of sites wanting to be involved, but sometimes it can be hard involving absolutely everyone. So as Andrew was saying, you do need sites that are capable of doing the trials, because you will be paying for each of those sites to get up and running.
And actually, Stuart, just talking about the EXPECT quant cardiac amyloid project, obviously cardiac amyloids are at a very interesting stage of development, and because there are therapeutics potentially available, it is something certainly worth looking at. If sites have EXPECT and they're interested in being involved in what you're doing, are you still looking for people for that, or do you want to comment on that, Stuart?
Yeah, so like Andrew said, my concept of the trial that we're doing, or the registry that we're doing, is that almost anyone can be involved if they want to be. At the moment, you need an EXPECT camera because it's going to be EXPECT quant at the moment. And the payment that we're planning to pay you probably saw on my slide, I said we were using the ALARA principle on payments. It's not going to be perhaps as large as one might imagine. But if you're interested in taking part in some sort of multi-centre process, really, I think it's a good way to get started. So our idea is that we will be sending our phantom around to sites who express an interest, and that will be paid for within the ARTnet grant process, and that we will have a relatively cheap way of getting information, your scans, from you to us, and that there will be a certain amount of action that you have to do at your end helping patients filling in quality of life questionnaires. So if you're thinking you're going to go into my study and make a lot of money out of research, that's not the case. But if you're thinking that you want to go into a multi-centre registry or a study and you haven't been in one before and you've got an expect camera and you might only do 2 or 3 patients, yeah, you're the people that we're looking for, as well as the large centres. So we're trying not to be one. You don't really need a lot of infrastructure, I guess is what I'm saying, to be involved in our trial. So it's taking us a while to get going, and that's partly because we're trying to keep funding at a relatively low level. But once we get going, we're encouraging anybody who's interested to contact us, and we'll consider them seriously.
That's great.
Yeah, okay. And I think from a therapy perspective, like with the therapy trial, we selected sites that had experience with lutetium dotatate at the time and also had on-site radiopharmacy with ability to perform HPLC quality control. So we had some prerequisites, and they also had to have a sort of prostate multidisciplinary team. And that network has now flown across to ENZA-p and UpFrontPSMA. And we have added some sites, and we do look to add more sites. And in that trial, ANZS-p, which is the other cooperative group who actually is the sponsor of the therapy trial, did send out sort of a national questionnaire to, literally, a large number of probably 50 hospitals that are part of their network to survey them. Do you have the necessary prerequisites? But there is opportunity to add sites, so always feel free to contact, I guess, the principal investigator of whichever study and see if it's possible. It may be something for ARTnet to consider, like a registry of hospital sites and their capabilities and areas of interest. So we're having a difficulty at the moment because there is one site that put their hand up for UpFrontPSMA, but it happens that they actually don't have a functioning HPLC machine. Now that's problematic because that's a basic quality control parameter that we cannot omit. So there are some stop-go parameters that we can try and sometimes work around, but can be problematic.
Okay, I'm just trying to see if there's any other questions that we haven't. Some of the questions, hopefully people can see the Q&A screen, and some of them have been answered live. There was a question to Andrew Scott about how long FIG may be accruing PET data for. I mean, I have to say the FIG study is very interesting, Andrew, because although FET is used clinically in many parts of the world, there really doesn't seem to be, as you mentioned in your talk, prospective studies really carefully examining the utility and being able to prospectively evaluate using parameters the interpretation of the scans. So that's a pretty interesting situation to be in, really, for that study.
Yes, it is, and I guess that was why we were successful with our grant application, fortunately. And it's been a journey, actually working through both protocol development and also the site selection, and due to start recruitment fairly soon. But we will have an awful lot of data at the end, and we're hopeful that it will be positive. But importantly, we've also tried to build a lot of science into this, so that the post hoc analyses of the images, the fact that we're getting tissue and blood, so we'll be able to look at genomic whole exome sequencing and RNA-seq in all of the tumour samples. I've actually got funding for that now. We will be able to look at circulating tumour DNA. We have funding for exosomes. So all of that additional analysis will be part of this too. So should be very interesting.
Yeah, be great.
And I think that raises the really important point that it's becoming increasingly clear that the quality of the data that you acquire is critical, and that imaging data really, if it's acquired really well within a multi-centre trial setting, it's just so valuable. And it's all about acquiring it well. And so doing these studies well. I mean, they're expensive studies to do because the expensive part is obviously treating the patients and enrolling the patients and managing the data. But as part of it, we perform a lot of different tests on the patients, including imaging. And I think it was also mentioned by Paul that, you know, perhaps looking at imaging data repositories or some means that we can perhaps after completion of the study and the main objectives of studies are completed, is there a way that we could still have that imaging data to be able to answer some other questions? And Paul, I think you raised that in your talk as one of the things that certainly strategically it is worth looking at. And I know NIF has an interest in this in part of their, I think, Australian Data Commons projects as well. And I think that there are some linkages there. I don't know if you want to talk about that.
Yeah, I think it's a complicated thing to achieve, and the National Imaging Facility, I guess, is very interested in doing this, but I guess it's been a little bit slower than we would have liked. There's a lot of different academic institutions involved, and of course they're very focused on the preclinical side of things. So I think as they proceed further, we're just keeping an eye on how they're going. As they proceed further, we will weigh in to make sure that they have clinical capability as well.
If I might say, and I think that this was touched upon by Michael before as well, I think in the next 2 to 3 years there's going to be significant opportunities because of the very rich data sets that have been generated through the trials that we've heard of and that are being conducted now, to be able to link into some of these major granting initiatives so that ARTnet and our community will be able to get funding to provide resources for these central repositories, which are going to be incredibly important. And there will be overseas collaborators that want to access it. There will be companies that want to be able to get access as well. There will be additional scientific questions in the future. So it is going to be something that we will be well positioned for, no question.
Yeah, I think that the great thing about these prospective multi-centre trials is you just collect high quality outcome data, and then you can go back and look at the images and explore them in a lot greater detail. So I think we're doing that in all these trials now. We've got these centrally collected imaging data sets which are well acquired using standardized techniques and good quality control around quantitative parameters. And now we can ask a whole bunch of, exploratory questions, and we embed this in the protocol and in the patient information and consent form so that we don't need to, go back and reconsent the patients. We let them know up front that their image data will be anonymized and stored and, may be used for future analyses to answer related or unrelated questions. And it's really a goldmine when you've got a 200 patient multi-center study with that data.
One of the things quite interesting to think about is who owns the data. So, and when it's collected through a different clinical trial group, it can become a bit of a tussle. And perhaps that's something that ARTnet should consider in the future, is owning a lot of this data.
Yeah, I think part of the key approach there, and certainly we're doing this in the FIG study, is that you have your publication policy and plans established up front. And that will ensure that all of the respective institutions and cooperative groups have a say in what actually happens. And certainly for the FIG study, ARTnet is part of it.
Yeah.
Yep. So I guess the question for ARTnet is, in the future, do you want to be able to sponsor trials? Or will you always be just an assistant and someone else will sponsor the trial? Because that is a big question that comes up increasingly. I was surprised to learn that Paul, Royal Brisbane and Women's Hospital, have never sponsored a multi-centre trial before, but it's a big issue, and it's it's an expense. Increasingly, that's an expensive process with a lot of insurance and legal oversight required for any entity to take overall responsibility for what's happening at all other sites.
And it's something that, so we do at Instant sponsor multi-centre trials. I think Peter Mac does as well, but our hospital's getting trickier about it, and it will change.
Yeah. And Peter Mac's actually doing the opposite. They're getting better at it. They've recognised that this is a opportunity. So the Office of Cancer Research has really grown a lot, and the legal department is getting increasingly expert at, at sponsoring multi-centre Australian trials, and they do that pretty well. And their next initiative is to be able to sponsor not only Australian sites but also international sites.
But I also think that, Stuart, your idea of a registration style trial that people can enrol into using REDCap, and that something like that being run by ARTnet is really, really interesting. We could have thousands of patients with fairly prescribed data going into those databases that could be extremely helpful. Or even in rare tumours, things like that, where we could get onto a database and add to it.
Absolutely.
I'm making notes, Louise. No, I think something we've discovered along the journey is there are plenty of opportunities, and that's exciting, to be honest. And I think hopefully I think it's remarkable what's been achieved so far, but it's also remarkable to think of what is still ongoing and can be achieved. This can only happen through goodwill, though, and I have to say, certainly when Paul and I and Andrew and other people have spoken to our international colleagues, I think one thing that's been very clear is that the environment in Australia and the collaboration between the different sites and investigators is incredibly strong, and is something that we should all be incredibly proud of. There's just been this real willingness to work together. I mean, I think population-wise, it is difficult to compete sometimes with, clearly with the US and Europe and big pharma, etc. But I think we've had a determination to show that, we can perform high-quality research, and we are really willing to work together. And we've had some fantastic role models in, Michael, Louise, Andrew, doing these multi-centre difficult studies that require large grant funding and leadership, but willing to do it cooperatively. And I know all of us at sites that are currently participating have really enjoyed being part of these studies and have benefited from the experience. And I think it's raising our levels everywhere. My lights just went out. And I think, there are probably... I'm in the darkness. And I think, because we conserve energy in WA, by the way, we're into energy conservation. They just turn off your lights. And I'm sure there are many other sites that are keen to be involved as well. And I think that is the next step for ARTnet, is trying to reach out to other sites and getting them involved. And, interestingly, FIG is actually at different sites to the prostate cancer ones. And I think as we cover different trials, there will be different sites involved. Stuart's study will involve different sites as well. And so that'll give a bit of broadness as well. I might have to turn my lights on. You'll have to keep talking amongst yourself. Like, someone will have to follow up with that while I turn my lights on. I can't see any other questions coming up in the Q&A. I don't know if anyone's raising their hands or if anyone's more better on Zoom than me to be able to see if anyone's got any other questions at all.
Andrew, can you comment on the overarching clinical trials network in Australia? I forget the title of the organisation that looks after all the clinical trials groups, of which ARTnet is now an official member. And I know ENZA-p, for example, gets significant funding from Cancer Australia or other sources. What opportunities do you see now that we have been successful with ProPSMA therapy, FIG, to just accelerate the ARTnet?
So the overarching clinical trials entity, it's called ACTA, which is the Australian Clinical Trials Alliance. John Salzberg's the president for life, I think. And so they've been able to negotiate quite good funding from the federal government. And you're right, ARTnet is now officially part of ACTA. In order to qualify for any funding, there has been a requirement which we're probably on the cusp of being able to tick every box for, which includes being able to independently be involved with the coordination or running of a clinical trial which has got to fruition and which has been published. So it is something that we'll be able to pursue over the next 12 months or so. One question which will come up is whether or not being able to sponsor the study is a prerequisite to get that substantial funding support. But that is something that we'll be able to start looking at.
How hard is it to sponsor, to become a sponsor? How expensive is that? Is that a big leap for ARTnet? Because it is It is probably an important leap at some stage.
You know, I mean, it requires an infrastructure. You know, you need to be able to handle all the legal, all of the quality, all of the data management. You need to be able to set up and run your own systems for, you know, case report forms, monitoring. You know, it requires a cohort of people being employed. You have to have the insurance, as Michael said. So you don't do it with just one person. You actually need a reasonable infrastructure to be able to do it.
Right.
Doesn't mean it can't be done, but
it's an aspirational goal.
Yes, yes, exactly.
And bearing in mind that ARTnet is in fact, you know, an ANZSNM and AANMS joint activity. So it's, you know, which is fantastic for our organisations in Australia that those, you know, two of our nuclear medicine societies work together so effectively to put ARTnet together. But yeah, so it then becomes a decision of what is the next step. And I think when we started all of this, we really weren't sure if it was going to lead to anything or not. And we're obviously incredibly pleased to see that it has. So then there is some decision making, I guess, about next steps as we go forward, which is an exciting problem to have. It's a good problem to have, really. So very happy about that. I'm conscious of time. I think it's great because we've still got quite a few attendees on the line who have hung in for the discussion session, which is really very fantastic, and thank you for doing that. I did see a hand go briefly up there, but for some reason, I'm not able to see who that is. I don't know if anyone else has been able to see any. If you've got a question, now's the time. Put your hand up. Otherwise, I'm going to see if anyone has any last comments on the panel that they want to mention before we wrap this session up. Does anyone have any burning last comments on the panel? Okay, so I'm just going to go back to my presentation screen. Okay, hopefully you're all now seeing the presentation screen. So just to really wrap up the ARTnet session, can I offer my thanks to everybody on the panel? Thank you for joining in the ARTnet session, and thank you for pre-recording your talks at very short notice, which I think did actually work quite well in the end. So I really do appreciate that, and thank you for a really interesting discussion. And really thank everybody who's on the line for joining in, and for the Australian community for supporting ARTnet. As I say, this isn't something that happens by individuals, it happens by everybody as a whole, and we're all incredibly grateful for that support, and we're looking forward to, you know, many other successful trials to come and lots of active discussions. Just a couple of housekeeping business before we wrap up. That hopefully people were able to obtain the QR code for this particular session. If not, I can tell you that the letters you need to enter in are EREZYL into Attendo. And I'm just going to now advertise our next set of sessions on our ANZSNM annual scientific program. So the next on-demand sessions, which will be launched on the website in the next day or so for radiochemistry. And so we'll have the following talks appearing on our radiochemistry section, and please go to your on-demand section of the ANZSNM ASM website. And also following this, our next live session will be the Shimadzu Award, where Giancarlo Pascali will be speaking in that particular live session, and that'll be on the 6th of August. We're also releasing onto the website some abstracts for our award sessions, and so they should be appearing on the website shortly as well. And so I think with that, we'll be wrapping up the live ARTnet session, and I'd like to thank everyone again for participating and wish you all safe times, particularly in these difficult times at the moment. So wish you all to be safe and well, and look forward to joining you for our next session. Thank you very much, everyone.
Thanks, Rose.
Thank you.
See you, everyone.